Interesting paradox in that organisms have this ability to make their own nitrogen, and yet we find many ecosystems across the planet, especially temperate and freshwater ecosystems, are limited by nitrogen, and oceans. So this question about why, when you have an organism that can make it, why should you ever have nitrogen limitation? And they address that. And I'd like to come back to that, so please look at that. And that's just one of, when I was studying this stuff as a graduate student, I actually took a class from Bob Howarth at Cornell, and this paper was huge in terms of my understanding of ecosystems, ecosystem processes, and ecological interactions that control biogeochemistry. So make sure you look at that. The Walker and Sayers, 1976, is another. So there's a mix of kind of new and classic papers on here. The Walker and Sayers paper also talks about nitrogen fixation a little bit, but it's mostly about how phosphorus, and I would say more broadly, rock-derived nutrients change through ecosystem development. And the Walker and Sayers hypothesis sort of argued that as you go through time, any ecosystem starts with all the nutrients really that it's ever going to have, and over time they kind of decrease. And it walks through why that occurs, variations in chemistry of nutrients and biogeochemical reactions over time that are important in defining this. The Pinuelos and others, 2012 paper, as a more recent paper, kind of a more synthesis paper, but looking at nutrient limitation is not just a academic question, it's really regulating the carbon cycle, right? And so that's why so much attention has been paid to how nutrients influence carbon cycling, because as we look, one practical application, as we look for ways to sequester carbon, nutrients will regulate how much carbon gets stored in ecosystems. The Elser et al. Paper from 2007, I've talked about quite a bit in here, and so that's the one where they actually did a big meta-analysis of how nutrients regulate productivity. These other two, so the ones that were assigned today, so the Galloway and others, 2008, really started thinking about nitrogen as an ecological disturbance, an agent of ecological disturbance, in that it creates a whole bunch of cascades. And the Schindler and others paper from 2008 really goes into that nicely, in talking about how nitrogen, because it's mobile in ecosystems, one atom of nitrogen can have multiple effects, right? Because it moves through ecosystems and eventually ends up in oceans. So today's fertilizer on an agricultural field is tomorrow's dead zone, pollutant that's contributing to dead zones. And that's a really nice, Dave Schindler is a Canadian scientist and has done some of this really cool work looking at nutrient impacts on lakes and doing whole lake experimental fertilizations to look at not only how nutrients affect productivity, but then how that cascades through food webs, right? Because once you change amounts or kinds of primary production, that can influence the types of organisms that are consuming those. And we're going to talk specifically about trophic dynamics next. I'll start it probably next time. But trophic dynamics is all about thinking about pyramids, right? Essentially, or food webs, and how energy and nutrients get transferred across different trophic levels, starting with primary production and then moving up through trophic levels. So I would urge you, and then the Hairston et al. Paper is the Hairston et al. In terms of trophic dynamics is the green world hypothesis, which is one of the most famous hypotheses in ecology. Why is the world green? We have all these herbivores capable of eating plants. Why does only 10% really of that energy get transferred to secondary or primary consumers? So I know you have a lot of stuff going on even in this class. There's an assignment due next week, but I would say, while we may not be addressing and discussing these papers in a really detailed way every week, I do see them as sort of a critical piece of building a bigger picture of what all the stuff we're talking about here. So make sure you at least come back to them when you have some time. They really are. I've done I spend a lot of time before this class every semester thinking really carefully about what sorts of stuff I think people should, at least as a foundation, should understand and be reading. So again, while we're not having quizzes and things like that, just recognize that it is a required part of I think, this course and the content of this course. So enough said there. But I want to finish, or at least continue, I guess, talking about the nitrogen cycle today. I finished with nitrogen fixation last time, and I wanted to come back to it. There were some questions at the end, and I wanted to make sure that everyone was on the same page here. This is a hugely important biological process, right? It allows, at least from a human perspective, it allows us to sustain numbers of people on this planet that we would never be able to sustain without it. Okay, so it is the most limiting nutrient, and this is the most dominant way of getting nitrogen into ecosystems. Okay, so I mentioned last time there's a symbiotic and free living components. I did look, Patrick, I sent you a paper this morning. I didn't send it to everyone, but it's a little unclear about heterotrophic nitrogen fixation in aquatic systems, but there seems to be evidence of nitrogen or of acetylene reduction. So to measure nitrogen fixation is almost impossible to measure directly because the atmosphere is 78% nitrogen, and so in order to measure nitrogen fixation, you're converting nitrogen gas to something else. And so if you're trying to measure a change in nitrogen concentration, it's really difficult to do that when the concentration of nitrogen is so high in the background. Okay, so no one really measures nitrogen fixation directly. We all use different techniques to do it. One way to do it is acetylene reduction. You guys heard of acetylene reduction? Acetylene is a compound that's also bonded, has a triple bond in it, and nitrogenase, the enzyme that fixes nitrogen fixation, will reduce acetylene to ethylene. So it does the same. It mimics the process of biological fixation, only using a compound that's not nitrogen. And so we use that as a proxy for N fixation. So we can measure the amount of acetylene converted into ethylene, and then we can sort of translate that to nitrogen fixation. Okay, so that's one way to measure it. That's done a lot on both of these, right? So you can put soil in a jar, add acetylene to it, let it sit over time, and then measure ethylene production. And that's one of the things folks in my lab do all the time, is use acetylene reduction to measure nitrogen fixation. The other way to do it is to look at the natural abundance of nitrogen. So because N fixation is fixing atmospheric nitrogen that has an isotopic, a del N15 value of 0, N fixers, if they're getting all of their nitrogen via the atmosphere, they should have an isotopic N15 value of close to the atmosphere or close to 0. So you can look at an N-fixing plant sitting next to a non-N-fixing plant. You could measure the isotopic values of those plants, and you would predict and often find that N-fixing plants have their tissue N concentrations are closer to the atmosphere because they're getting some of their nitrogen from the atmosphere. The problem is that N-fixing species don't exclusively rely on the atmosphere for their nitrogen. They can also get it from the soil. So they mix, their source of nitrogen is mixed. But natural abundance is used. So the natural abundance just means what is the isotopic concentration or the isotopic value of nitrogen in leaves, for example. And that's one way of detecting N-fixation. It's harder to ascribe an actual rate of N-fixation using that technique. What you can do is use a labeled form of nitrogen. So if you take a plant in a jar and you add nitrogen that's labeled, so using this nitrogen is really awesome to study because it has a stable isotope. So if you, N15 being the stable isotope, most of the nitrogen in the atmosphere is N14. There's a stable isotope N15, and you can label plants with N15 and then look for that in their tissues. So if they're taking up that labeled nitrogen, it's easier to follow a labeled form of nitrogen than it is to follow just natural nitrogen from the atmosphere. Okay, and so that's how you're measuring these things, and these things have been measured in sediments. So using the acetylene reduction technique, they've measured it. Seems to be lots of rhizosphere bacteria, like in typhus I was reading about, and some salt marsh grasses. There's lots of N-fixation in salt marshes. What's a little unclear is what the actual organisms are that are doing it, right? So if they are truly heterotrophs or if there are some sort of photosynthetic, autotrophic end fixers. But my guess is that in sediments, there's where you have high carbon, low nitrogen, and where there's oxygen, you would find heterotrophic end fixers. But it would be interesting to dive into that a little bit more deeply. But I did send you a paper this morning, and if others are interested, I'm happy to share that. But it was talking about end fixation in saltwater and freshwater environments from limnology and oceanography. Okay, so the free living part is ubiquitous, I would say. You find it in soils. You find it in canopies, like on leaves and trees. If you take a leaf and we've done this too, you take a shotgun and shoot leaves off of canopies and collect the leaves and put them in a jar with acetylene, you would find that there's epiphytic bacteria on the surface of leaves that are fixing nitrogen. You find it in decomposing litter. You find it in wood. Okay, so in ecosystems where there's lots of carbon but it's difficult to break down, those are the types of environments where end fixers are favored because it's a high carbon, low nitrogen ratio, and so those are where you would expect lots of free living end fixers. Okay? Symbiotic is much more, I guess, patchy on the landscape. Symbiotic is typically people think of legumes, okay, with the rhizobium bacteria that legumes have in nodules. That's only a very small fraction of the end fixation that's actually happening symbiotically. And there's very few symbiotic end fixers in general, but legumes especially outside of the tropics. Okay, there's lots. The Fabaceae, the legume family, is a hugely abundant family, but most of them are in the tropics, especially the trees. Okay, so and this one, the controls on symbiotic end fixation, this one's pretty easy to measure, right? We know, for example, that end fixation is inhibited at high levels of nitrogen. So when nitrogen is available, they turn off. That's a facultative process, not an obligate process. So plants can control it. So when there's a lot of nitrogen available, they can shut down nitrogen fixation in the same way that in high nitrogen environments, if you add nitrogen to soil, you would find typically that rates of end fixation would go down. So there's this trade-off between supply and then the ability or the rates of nitrogenase or end fixation activity. Okay, and there seems to be something here that's really important too. And why, one of the reasons why it's speculated that nitrogen fixation isn't more common is that phosphorus is needed in high concentrations, as well as things like molybdenum and vanadium and chromium and iron and these weird trace metals that sometimes aren't really abundant. And so if you don't, because of the importance of stoichiometry and the ratios of elements, if you don't have enough of things like phosphorus, then nitrogen fixation is suppressed. And similarly, if you do, there's been lots of studies in the literature fertilizing with phosphorus and trace metals, and when you do that, end fixation cranks back up again. Okay? So, and that sort of argument has been framed as there's no real such thing as nitrogen limitation, it's other nutrient limitation in disguise, right? So if you have phosphorus that's in low concentration, it comes across as nitrogen limitation, but really if you add phosphorus, then nitrogen limitation cranks up. Okay? And that's this, sometimes in the literature you'll see discussions about proximate versus ultimate nutrient limitation. Okay? So ultimately in that case it could be phosphorus that's limiting, but proximately it looks like end limitation. Okay, does everyone get this? Just briefly, this isotopic question. We got that? Is that clear now? Okay, good. Okay, so it requires tons of energy, and that's another part of this, right? The energy in the form of ATP, right? So for every mole of N2 fixed, it requires 16 moles of ATP. So that's a huge phosphorus and energy demand there. And so it makes sense that you don't find it in areas like, for example, end fixers, symbiotic end fixing trees are most common early in primary succession, and then they tend to die out. How come? could, they're definitely, so they're using lots of phosphorus at the beginning to make the energy, but why would that essentially set up a situation that disfavors them later in succession?
Limitations in succession.
Exactly. So they need a huge amount of energy to drive the photosynthesis, to reduce all that phosphorus, or all of the ATP to run these reactions. And so when the canopies close, so early on, they tend to be fast growing. Early on after disturbance, nitrogen tends to be fairly limited in availability. So they capitalize on that and grow up, and like in canopy gaps, for example, they tend to thrive, and you find lots of end fixers early in succession. Over time, when canopies start to close, shading, right, lower rates of photosynthesis, then they tend to be at more of a disadvantage, and they tend to be eliminated. You don't find them they're not nearly as common in late succession as they are in early succession. That also probably has to do with the fact that nitrogen inhibits end fixation, because as they fix nitrogen and as they're growing in early succession, they're adding nitrogen to the soil, which tends to have a negative feedback on nitrogen fixation. Okay, but it's really energetically expensive, so you don't tend to see it, again, especially in late successional forests. We have some early successional end fixers in the northern hemisphere, in the northern temperate forest. Black locust is one of the only ones, but black locust is a successional species. It tends to die out and is not a sort of late succession, mature canopy species. Only select bacteria for legumes. It's only one genus of bacteria, the Rhizobium. Okay, so Rhizobium bacteria associate with legumes. The actinobacteria associate with some of the actinorhizal end-fixing species, which I'll talk about in a few minutes. Nitrogen fixation is inhibited by oxygen, so nitrogenase, the enzyme, is highly reactive to oxygen. It degrades it, and that's because it's got all these metals and things in there. And so this is much more common in intracellularly. Organisms have evolved ways to remove oxygen from the site of end fixation, but even ecologically. So if you go around, there's been some really neat studies on Barrow, Colorado Island in Panama that show that end fixation is actually greater right near the shore. So why would that be as it relates to oxygen? So if you start from the shoreline, so Barrow, Colorado Island was formed when they built the Panama Canal. So most of the Panama Canal is just a big reservoir that they dammed, right? And Barrow, Colorado Island was just part of the rainforest, but when they submerged the lake, Lago Gatun it's called, then it created an island. So as you go from the shoreline upland into the forest, rates of end fixation decline. And it's thought that actually has to do with the fact that in saturated kind of shoreline environments, lower oxygen concentrations, and so plants have to actually work less hard to get rid of the oxygen because the sediments are more saturated closer to the shoreline. And this is really important so that this process can go up or down depending on nitrogen availability. And the facultative nature of end fixation means that plants don't have to do it all the time, because if they did, they would never survive outside of very restricted environments. But they can turn it off. So you do find end fixers that fix early on in their lives, and then as canopies close, for example, you still find some mature, in the rainforest especially, you find mature end fixing trees, but they just aren't fixing nitrogen anymore. Okay, so they control this process. The bacteria, Rhizobium, are in all soil. They're just free living bacteria. The plant basically turns on something that signals the bacteria to come inside and make nodules from me, right? So, but that's really sensitive, not just to nitrogen concentrations, but to the concentrations of other elements that are needed. So here's some of this, like in the fall in your garden, you get clover oftentimes, or in your yard. If you dig that up, you would find this is what it looks like, and you find these nodules here. So these are the site where, and like your clover is a legume, so it would have rhizobium housed in these nodules. Okay, and these nodules are again oxygen free, and that like the leg hemoglobin is what gives these things their reddish tint, but then it's moving oxygen away from the nodules where the end fixation is occurring.
So is it known, I mean, you say they can shut it on and off, the end fixers can, but is it known in the literature, that signal, you say, like what it is?
Totally. Yeah, there's books, this is agriculture, right? So agriculture, anytime you have something that deals with food, it's usually pretty well studied. And so end fixation and the signals and the biochemistry of it is really well understood. I don't know it very well, but it is very well understood. I'd say much more so than the ecological controls. The sort of physiology of end fixation is really well understood.
Corey, is the iron deficient or iron limitation related to why that's red?
No, so the leg hemoglobin has iron in it, right? Just like our blood, the hemoglobin molecule in our blood has iron in it. So you can have in low iron soils, it can inhibit end fixation because of the ability to, or the inability to produce that. But yeah, that is what gives that sort of pinkish tinge, is that leg hemoglobin. And it's exactly the same, well, very, very similar molecule to the ones we have in our blood. So this is a sort of a cross section of one of these nodules, and this would be the rhizobium in here. So they're getting carbon from the plant, and they're in this anoxic environment. They're taking nitrogen gas and reducing it into a reactive form of nitrogen. Okay, and this is, again, just shows you, and this is really clear. If you take a clover plant from your garden and slice one of the nodules open with a razor blade, it just instantly goes to red. And it's just the reaction oxidizing, the oxidation of iron, right? Iron 3 is sort of that reddish rusty color, and that's the same sort of thing we're using here. Yeah, so when a plant is essentially turns on end fixation, it creates these infection threads and sort of invites these rhizobium that are in the soil inside to create these nodules. Right? And it releases them, and then it would sort of seal this off inside of the nodules. Okay? Mostly, again, to keep the oxygen out of that. But if you're interested in that, I can send you some. This probably talks about it at some level in the book, but if you wanted more detail, I could point you in the right direction. But this is it's thought, again, to be largely controlled by the plant. Okay, I'm not implying that there's no sort of interaction here, but most of this, whether this is happening or not, seems to be controlled by the plant. And oftentimes you can go into plants and find dead nodules, right? Or just they get dried up when they're dead. And a lot of that just has to do with seasonal variation in environmental conditions as well. The plants just decide, like, nope, this is not enough energy return on my investment, so they turn it off. There's other ones. We talked a little bit about allness in the Pacific Northwest. Allness being a very much of a disturbance colonizing species. So you find them a lot after landslides in the alpine here. Avalanche slopes have alder on them, and those are end-fixing species, or end-fixing members of allness. Other ones around too, desert in Ceanothus, in the chaparral communities, lots of end-fixers. Yeah.
So would you think that one of the reasons why you also see alder a lot in more like riparian areas and wetlands and streams and stuff, because of that oxygen
Could be.
for helping nitrogen fixation?
It could also be sites where N is, because it's wet, it's constantly being leached out, right? So because N is mobile, it's constantly being moved, right? And so it might just create riparian areas, may actually be even more N limited than some of the upland areas, right? Because you're constantly getting movement of water. Okay, so and also where you would tend to see deposition from upland systems of the other things that tend to limit end fixation. So it's a good, we could test that, right? I don't know, but it seems like a really obvious hypothesis.
I mean, you could also argue that they might not even be fixing in, because wherever the groundwater is coming down, it has nitrate in it, and that way they're not limited before it hits the stream, because they're right there acting as a buffer. So they might not even be fixing that.
Possibly, and I don't know, what are the species that tend to colonize riparian areas of alder? Is it green alder?
Incana,
red.
right?
So usually the sort of world record rates of end fixation are red alder, like 150, 200 kilograms of nitrogen per hectare. So they crank usually, but it could be that they're not all fixing at high levels or even at all, I don't know. But they tend not to be, these are much more common actually in temperate zone, these actinorhizals. So this is alder and ceanothus. They build, they're what's known as actinorhizal end fixers. So they build nodules, but it's not Rhizobium in them. It's actinobacteria, and they're slightly different. And this form of end fixation is pretty broadly distributed throughout the plant kingdom. So you find lots of examples among different families of these actinorhizal end fixing species. The legumes, the Fabaceae are a very specific and very common type of end fixer, but it's restricted to the Fabaceae family. Yep.
Is that the ceanothus that's around here as well?
It is. Yep. So we have ceanothus around here, and it's a pretty active end fixer. There's quite a few shrubs around here that have this actinorhizal type of end fixation. I think some of the ericaceous species, I don't know if huckleberry is, but there are some, I keep learning about them. This summer I've learned about a couple of new ones, and I've always been curious, because we lack lots of legumes around here, where do our ecosystems get their nitrogen? But there's quite a few shrubs that occupy this, or that conduct this sort of end fixation, that are pretty common around here. But yeah, this ceanothus is definitely, ceanothus just being a genus, right? So probably lots of members of that. So Frankia, you'll see Frankia as the genus of actinobacteria that is common in alders. And they're pretty obvious when you see them in soils. They're harder, so the nodules of the Fabaceae, you can actually crush them, right, with your fingernail. These things tend to be much harder. I've seen there's an alder is very commonly grown in the tropics in plantations because it's really fast growing. And I was at an alder plantation once in the high elevation site in Costa Rica that had nodules that were like the size of lemons, just giant nodules in the soils. And they crank lots of end fixation. But they're clustered in these, they do look like nodules, but they're different. Okay, they're occupied by a different bacteria, and it's not exactly the same physiology that I was talking about with the legumes. So Alnus, for example, alder has Frankia, right? That's part of what their end fixation, their style of end fixation, right? Or their method of end fixation includes Frankia, but so would Ceanothus. So the Frankia would be the genus of bacteria that's fixing, but it's across lots of different species and across lots of different families. Okay, and then these guys, so just a couple of examples. Azotobacter, a heterotrophic end fixer, lives mostly in soils. Nostoc, I think I mentioned Nostoc, is a cyanobacteria that's common, in soils, in wetlands, in all sorts of aquatic environments. This one, Nostoc, is a photosynthetic end fixer. And it has these really cool structures called heterocysts. So these would be the normal cells, and then these heterocysts are these cells where the end fixation is happening in Nostoc. And that's where essentially the place where oxygen would be actively removed to prevent oxidation of the nitrogenase enzyme. Okay, so where would you expect to find the most abundance of these? They're kind of everywhere. But think about the ecological conditions that I just talked about that would tend to favor them, right? So if there's lots of carbon energy, but you can't use it because there's not enough nitrogen, that tends to be the areas where you find it. So decomposing leaf litter and decomposing wood, there's tons of carbon there, but it has a really high C to N ratio, and so heterotrophs that aren't fixing N can't essentially balance the stoichiometry of what they're eating with themselves, and so those are the areas where you tend to find higher rates of free living N fixation. High carbon, low nitrogen environments. So sediments and soils and decomposing organic material. So oddly enough, for as important of a process as this is, our understanding of kind of the rates and magnitude of N fixation globally is pretty poorly understood. This was actually when I was a graduate student, a master's student at Cornell, we had a class where we did together like a group project, and in that group project we did N fixation. Like, let's look at the literature and find rates of N fixation in different ecosystems. So there were like 8 or so of us. We each took a system and went into the literature and found 10 papers and then put them all together and published this paper, which for the first time tried to say what is global N fixation. Okay? And so we looked at both these free living N fixers and legume N fixers, and we acknowledge the fact that lightning contributes something, but the vast majority, more than 99% of the nitrogen comes from biological N fixation. And what you actually find is that N fixation, if you plot rates of N fixation against a number of environmental variables, you find good relationships. So climate, for example, explains a lot of N fixation. Why? How would you expect N fixation to vary with climate? Or looking and thinking about the patterns here. So you tend to see the highest rates here, and it gets lower as you go towards high latitudes.
Decomposition higher, it means more carbon.
Okay, so carbon. So just carbon alone, right? If we know we need energy to fix N, you might predict that higher rates of N fixation in areas where you're getting lots of energy production. So sunlight, and actually the relationship we developed from this that helped us produce this map was evapotranspiration again. So evapotranspiration is energy, sunlight, right? Water, all of these things, like an integrated look at all of these things. And so if you plot N fixation rates against AET, you find a very nice relationship. And so then we used, using a global model of AET, we generated this that showed rates of N fixation. Okay, this basically showed that this number is pretty small, but because they're everywhere, it's an important contributor. So free living N fixation rates are generally much smaller than you'd see for symbiotic N fixation. But if you multiply that, these are only common in sort of patchy areas. Okay, so this one ends up being very important. These are some of the ones that we looked at with, like the alder, right? So when those things, when all things like alder are present, they can crank, but they tend to be a very low distribution or poorly distributed. And in the end, this sort of suggested high rates of N fixation in the tropics, again, consistent with the observation that that's where most of the legumes are, right? Consistent with the fact that there's lots of energy, right? One of the sort of confounding things is that those are the systems that tend to have limitation by other elements. And some of this was from 1999. This was the very first paper I ever published as a graduate student. And there's been a lot done since then, but the reality is, like this this is basically, the patterns, the numbers are kind of changing a little bit, but the patterns have remained largely the same. And it's still based on very poor data. For places, for big parts of the world, people just haven't measured it. And I just wanted to mention briefly some of these cool things about nitrogenase as an enzyme. So this is the enzyme that takes N2 and converts it into ammonia. It has all these weird things as a part of it. So molybdenum. Like, who knew that you needed molybdenum to grow? Vanadium. Yeah, and it's just, it tends to be, when end fixers have this, they need a lot of nitrogenase, and they're constantly investing energy into producing it, but then also keeping it from being degraded by oxygen. So whether you're talking about legumes and rhizobium or actinorhizal end fixers and frankia, it's still nitrogenase. And the type of nitrogenase can vary a little bit, but this is still the enzyme that's doing it. And there's actually been some cool sort of phylogenetics of end fixation that suggests that actually this was very common at a sort of a root level in organisms, but that some, like in the phylogenetic tree, some organisms, groups of organisms, lost the ability to produce nitrogenase over time. So that's why it's sort of patchy around the phylogenetic tree. Okay, so this was just sort of to articulate this more clearly. So the challenge of understanding N limitation in an N-fixing world is framed as follows. N limits productivity, composition, and dynamics diversity of many ecosystems, and that they respond when N is added. Okay, we know this. You add nitrogen to most, this is why we add so much nitrogen in agricultural systems, because they grow more. Okay, so those are, by definition, N-limited systems. And basically, I've just told you that free-living N fixers are ubiquitous, right? And that there's, trees should be able to capitalize on this too. So organisms with the capacity to fix atmospheric N2 are ubiquitous and diverse. So how can you ever have limitation by nitrogen, right? In light of the fact that there's an endless supply of nitrogen in the atmosphere that could be fixed, right? And the biological capacity to make use of N2. So how could this ever exist? Did anyone read the Vitusek and Howarth paper?
Other nutrients?
Okay, so yep, we've mentioned this a little bit. So I like the way they phrase it, the other nutrients in disguise, right? So it could be molybdenum or phosphorus. They kind of focus on P as probably one of the big limiters of N fixation. What else could limit N fixers as plants in ecosystems?
Iron.
Okay, but that's other nutrients, right?
Climate?
Sorry?
Climate?
Okay, so how would that work?
Like at high latitudes, cold temperatures.
Okay, perfect. And so there's physiology of nitrogenase that looks like it's a hump-shaped relationship. So if you have temperature and nitrogenase activity, it kind of looks like that. Okay? And so there's a temperature optimum. And so once you get down here, then temperature can actually constrain rates of nitrogenase activity. So it's a very, very temperature sensitive process. And so as you get into northern latitudes, cooler temperatures can certainly constrain or inhibit rates of N fixation. What else?
You were saying before, like light availability.
Yep. So resources that would include, so light and nutrients, climate, any other ecological factors.
Herbivory?
Disturbance.
Okay, so herbivory. What do you mean herbivory? how would herbivory tend to have a disproportional effect on end fixers?
So if end fixtures tend to be integrated succession systems, for example, they might actually be, there might be fewer plant species for herbivores to prey on, and we could get to a higher rate of consumption.
Okay, so early on, it just might be the end fixers are what's around. What about in more diverse systems where you have a mix?
Don't a lot of herbivores preferentially eat like high nitrogen content plants?
Don't you?
Yeah, with deer and all the ceanothus.
Yeah, I mean, that's why we cultivate legumes, right? Legume cultivation is all about quality of food, really. I mean, it's a high nitrogen, high protein, nutritious types of food. Herbivores do the exact same thing. They preferentially are gonna graze things that are gonna have high nutrient content, that are gonna be higher food quality. And so in ecosystems where you have end fixers, they tend to be preferentially grazed. Have you ever seen the elk in the alfalfa fields in the summertime? Like, they're not stupid, right? Like, there's just bleeding nitrogen out of alfalfa as an end fixer. It's the legume family, and you know, that's where they would tend to concentrate. And so there are ecological factors that could explain this as well. And that largely has to do with the fact that herbivores, as end fixers, are just all other things being equal, herbivores may preferentially graze end fixers. Yeah?
If we're looking at like the hierarchy of impacts upon nitrogenase, is temperature less important?
Good question. So this is all really super interesting ecology of end fixation. Because if you have multiple controls, you have to think about all of them, right? So, in what sorts of environments in what sorts of environments would you expect temperature effects to trump nutrient effects?
Well, you see, I guess what I'm kind of confused by is the tropics have the highest fixation of nitrogen, and if it's not a very strong control, that makes sense.
So just recognize that this is probably 50 degrees C here.
Never mind.
Sorry, I didn't write numbers on here, but the temperature optimum, the tropics are well below the temperature optimum. But the point is that there's biophysical and biogeochemical controls on processes, and that either one of them may be more important depending on where you are. So if you're in systems where you have lots of water and it's warm, then that's where you would expect the nutrient controls to have a bigger effect. But if you're in a system that's dry and cold, then those are where you might expect the biophysical constraints to have a bigger effect. And that's actually, like, people are doing research on that right now, trying to tease that apart. Like how, as you go, for example, across temperature gradients when rainfall is abundant, you would expect that the temperature controls are much more important in cooler places and that the nutrients don't really matter because temperature is suppressing rates of nitrogenase. But as you go up in temperature, then you start to kick in these other controls. And that's the way lots of processes work, right? We talked about that a little bit with NPP, right? So nutrient effects on NPP probably aren't going to be very important in systems that are freezing cold or really, really dry, right? But once you're in climatic zones that favor those processes, then these other controls start to matter more. So have a look at that paper. That's sort of the gist of it. But there's been some really cool studies looking in lakes, for example, and when you have lots of end fixers in lakes and how zooplankton numbers crank up, right? And so these really cool ecological effects, especially grazers in aquatic systems, because end limitation is very, very common in aquatic systems. So why aren't end fixers more important? Well, when end fixers crank up, you tend to really crank up grazing and zooplankton that are eating them. But they do a really nice walk through a bunch of different examples and case studies, and it's all really just kind of a hypothesis, and then they present some data in support of them. But it's been, I'd say, certainly for my research, like I've had multiple, written several unsuccessful proposals as part of this, some successful studies, but trying to understand this paradox, because it really doesn't make very much sense. And really, I've focused more on nutrient controls. But usually, like if we went out into a natural system here and added nutrients, we would find an increase in N fixation, just because that tends to be here, especially like in like riparian areas or areas where water is available. It's the nutrient controls would matter. And not trivial, right? So I'm actually reviewing a manuscript right now submitted to Nature Communications or something, where these folks are arguing that as a result of climate change, N fixation will increase, right? So they're saying, well, it's going to get warmer, and so this is going to go up. Right? And so what that means is that the terrestrial biosphere in the next 100 years is going to store way more carbon because of nitrogen fixation. But the point is that this is sort of a theoretical, and if you look, this is from lab-based assays looking at temperature effects on nitrogenase. That doesn't necessarily mean if you warm up the world, nitrogen fixers are going to crank out all way more nitrogen. And that's partly what this is getting at, right? You can't predict simple responses of N fixation based on something like temperature, because there's ecological effects, there's biogeochemical effects, all these other things. Okay, so other inputs. I spend the most time on the biggest and I would argue most important one. There's some evidence, especially in certain areas where you have sedimentary derived parent materials, that geologic inputs of nitrogen can be very important. This set of studies, Dahlgren in 1999, and then there was one, Scott Morford and others in Nature from just a couple of years ago, where they actually argue for that the importance of geologic N has been greatly underestimated. Okay, so it doesn't seem to be the source of the majority to N to most ecosystems, but it can be the source of N to some ecosystems. Atmospheric nitrogen deposition is another one that's become a huge, I shouldn't say huge, so if we're thinking huge, N fixation is huge. This is becoming an increasingly important source of nitrogen to ecosystems, nitrogen deposition. The patterns suggest kind of the controls here. And where's all this nitrogen coming from? I think we talked about this last time, right? Some of it's agriculture.
Industrialized areas.
Yeah, and mostly like fossil fuels, right? So Eastern Europe, the northeastern United States, right? The heart of kind of production, like a lot of it was coal-burning power plants. Okay, but automobiles contribute a significant source of N to the atmosphere. A lot of this is probably agriculture here. Okay, this is from the mid-90s. So, and if you look at predictions for like 2050, nitrogen deposition rates are predicted to increase like 4 or 5-fold. Do you have a question? Okay, so if you add up all of the sources of N, this is actually a fairly modest one, but it's becoming more important, and it's reactive nitrogen being added on ecosystems that wouldn't ordinarily be there, right? And in places where you wouldn't necessarily expect high rates of N fixation, for example. So this is what I mentioned last time. The source of this is actually ending up over here and there's big high rates of deposition happening just from downwind transport of this nitrogen over China, Southeast Asia. Some of this, when I'll talk about some of this is largely being dealt with in Europe, and for the most part, rates of N deposition in North America are starting to decline, largely because of the Clean Air Act, but it's still big, especially sort of in small sites. So like in the Colorado Front Range, there's a big urban center, and that N sort of that's emitted to the atmosphere kind of sloshes around in that Front Range, and then between the mountains and the city, there's a big N deposition, local N deposition hotspot in the Front Range of Colorado. So here's how things have changed. So the number I showed, remember I said I showed you the map from the paper we wrote, and that we basically added up all of that nitrogen in that big map and came up with a range of estimates with a ton of uncertainty. So anywhere from like 75 to, let's see here. So natural end fixation is thought to be about 100. Right, so here's the range of natural end fixation estimates. So from something like 75 to 150. That's how much natural biological nitrogen fixation roughly is thought to add to the world every year. We cultivate legume crops, so legume crops are not considered part of natural end fixation. It's a new kind of nitrogen, so that's mostly soybeans, all of the legume crops, the pulse crops. So that adds another 50 or so, and this is in teragrams of nitrogen per year. Okay, so legume crops bring us up another 50, fossil fuel, right, another bit. The point of this is that basically the total amount of anthropogenic end fixation right now is thought to exceed natural end fixation. So humans, most of that is from the production of end fertilizer. So all of the human cultivation, production of fertilizer, all of that combined fixes more nitrogen than all natural ecosystems combined every year. So we've more than doubled the amount of nitrogen cycling around in the planet. And that comes with all sorts of costs, everything from eutrophication of aquatic systems, like I told you last time, and I was a part of a working group at the National Center for Ecological Analysis and Synthesis as a postdoc, and we looked at the effects of all this nitrogen on human health, right? So anthropogenic nitrogen in cities has huge effects on respiratory diseases like asthma. End deposition in aquatic ecosystems has been shown to favor some aquatic diseases, so like cholera and malaria actually does much better in eutrophic than oligotrophic waters.
E. coli does too.
E. coli, exactly. Pfiesteria, you guys have probably heard about some of the pfiesteria outbreaks in systems downstream of like pig farms and stuff, and that's a very much of lots of these pathogenic bacteria love eutrophied waters. Okay, and then things like pollen production are also highly regulated by nitrogen availability, and so allergies and allergens, ragweed, for example, produces way more pollen when it's grown in high nitrogen soil than low nitrogen soil. So there's a whole bunch, and this is the cascade of ecological effects of something like nitrogen. Okay, that's become not just, again, just an academic question about limitation, but an element that is causing massive problems, global scale problems, and real economic impacts. Also, one I would argue, as opposed to maybe climate change and carbon and regulating carbon, which is really hard, this one's actually way easier to deal with, I would argue, right? We use too much fertilizer and we apply it in the wrong time and in the wrong place and in the wrong quantities. But you could manage that better, right? We could figure out ways of, most of the reason people add so much fertilizer is just because it's cheap. Like, why not? Just dump it on. But that was before some of these effects started, becoming very obvious. And this is a problem that has gotten much less attention. You guys maybe have not even really thought about nitrogen as an environmental problem, but I would argue it is having at least the time course over which these changes have taken place and the effects have been much shorter and more recent than the effects, like the longer term effects of climate change. Okay, so there's real opportunity for thinking about how to reduce some of these ecological effects that the nitrogen cycle is having. We just have to do it. Here's an interesting one. Yeah, go ahead.
Well, it's kind of a I'm from a small town in Vermont, and there are lots of Holstein dairies.
Yeah.
And every year it's in the news that farmers get anxious to spread the cow manure in the spring, but you can't do it before the before the ground is thawed, because there will be overland surface flow if there's a rain.
Exactly.
So it's a big problem there that is actually, considered by a lot of people.
Well, and people add fertilizer in the fall sometimes, right, for winter wheat and stuff, and it makes sense because you're like, well, when they wake up, they're gonna need it, right? And the fields are often muddy in the spring, and so it's harder to do, but it also makes it much more susceptible to leaching in the fall when you do that. One of the upshots of this paper was, how do you deal with some of this end fertilizer stuff? And, well, I mean, maybe I can't ask a question. What we showed in here was that much of the end fertilizer was actually being used to fertilize things that animals eat, right? And so one of the easiest, if you want to put huge quotes around that, or one of the most obvious ways of dealing with some of this nitrogen issue would be people eat more plants in their diets, right? Because animal consumption and a lot of this nitrogen fertilizer, for example, corn, right? And using fertilizers to fertilize plants that are feeding animals that are feeding us, if you cut out that part of it, this nitrogen problem, a lot of it can be solved literally overnight, right? Just more plants, more of like what we call a Mediterranean diet as opposed to like a North American diet. So if you're not a vegetarian, but you want to help with this, just have plants one more day a week and you're making a big difference.
Well, it seems like you could just eat the right type of meat too. Like you just don't eat factory.
Like hunted meat or something? Yeah, totally.
It's not relying on this.
Yeah, absolutely. I mean, that's what's awesome about the Montana carnivore diet, right? Is that it doesn't really, you could argue at some level you might be rationalizing it, but it certainly doesn't require the sort of energy, and this is not just nitrogen, this is energy. How much energy it takes to produce, to sustain diets that aren't plant-based is just enormous, right? So by reducing, meat consumption just once or twice a week, you help with this problem. And I'm not here to preach to you, and I'm just saying it's an ecological problem that has real societal implications, right? This is something that's linked to big changes that you could make big dents in, or as a society we could make big dents in just by simple choices. Whereas carbon, right, it's a little harder, right? Like we can't just stop driving cars. We can't just stop using fossil fuel heat, right? It's not as straightforward. This one is a little more straightforward. There's actually another aspect of this that I haven't mentioned is the nitrogen effects on the climate system. And Dan was in my climate change class and I talk a lot about this. But nitrogen has big effects on the climate system as well. And so for the first time, the National Research Council did a carbon climate assessment for North America. And for the first time last year, or a couple of years ago, they actually had a nitrogen cycle side of that carbon climate assessment. Okay, because nitrogen, it influences carbon, right, through the interactions that we've talked about, but also there's a really important trace gas produced as part of the nitrogen cycle that a lot of this stuff, right, if it's not actually taken up by plants and it's not leached into even if it is leached, eventually it could come out as nitrous oxide, and nitrous oxide is a really important greenhouse gas. Okay, so those are our inputs here, and so now I want to start talking about internal transformations. As people who are, well, maybe not everyone going to think about end fixation or the nitrogen cycle after they get out of this class, but most of you at some level will. And so I want to introduce you at least to this cycle and the processes that are important. So end fixation, and I've simplified things here. I'm saying N2 is being fixed by plants, and that eventually enters the soil organic matter pool. Okay, soil organic matter pool. The soil organic matter pool then becomes susceptible to decomposition and the start of this cycle, or I guess the next step of the transformation of nitrogen. So nitrogen that's fixed in plants and enters as soil organic matter, decomposition mineralizes that nitrogen. Okay, so when plants decompose organic matter, ammonium gets mineralized from that. So mineralization, jargony term, but it's decomposition. So decomposition produces ammonium, and that process is called mineralization. Okay, once nitrogen is mineralized, and if you go to any soil around that's not an agricultural soil that's been fertilized, these concentrations of this stuff are really, really quite low. You can measure it, and it varies from system to system, but this stuff is pretty low supply. Okay, and that's because lots of stuff are competing for it. Plants that aren't nitrogen fixers, they need that. Okay, so most of them can only get it after it's been mineralized. So usually if you measure net rates of nitrogen mineralization versus production of ammonium, rates would be really high, but this pool stays continuously small. So as soon as it's mineralized, it gets taken back up again. It can be taken up by plants, roots, or immobilized by microbes. So microbes that are in soil need nitrogen just like the plants do. So after it's decomposed, microbial organisms can take it back up too. Okay, so there tends to be a very tight relationship between mineralization and immobilization, and sort of the net amount of nitrogen is actually produced is actually quite small, or the pool that you can measure is actually quite small. Once this ammonium is produced, most of the sinks for it are biological. It can be lost from the system, but as we talked about a lot in the first part of the class, this is a cation, and so ammonium can be sorbed onto exchange sites. Okay, so to measure this, we actually do extractions with salts that releases ammonium off of those exchange sites. And if this is on an exchange site, a plant or a microbe can get it too. Okay, so actually quite small amounts of ammonium lost from leaching. Most of the form of nitrogen, or the most dominant type of nitrogen leaching is ammonia, or is nitrate, because it's a negatively charged cation or anion. So end mineralized to produce ammonium, either taken up by a plant, immobilized by a bacteria, or it can get nitrified. Okay, so nitrification is the process where, and this is actually two sets of microorganisms, two genera of bacteria that do this. They convert ammonia first to NO2 and then a second step to NO3. And that is an energetic, that's an oxidation reaction that releases energy. So these are heterotrophic organisms in soil that are chemoheterotrophs, and so they're getting their energy from nitrification. This is kind of if you imagine this is a pipe, so we have the pipe going, and ammonium is going in one end and nitrate is coming out the other end. It's been referred to as a leaky pipe because this nitrification process can actually produce some other things, NO, nitric oxide, or N2O, as a byproduct of nitrification. Okay, so, and here's our greenhouse gas, or one process in the nitrogen cycle that produces a greenhouse gas. So nitrification converts ammonium to nitrate, and in the process, there's this sort of incidental production of nitric oxide, which is can aerosolize and part of, create smog in the presence of ultraviolet light, and then nitrous oxide, which is the third most important greenhouse gas in the atmosphere. After CO2, what's number 2? You know? Methane. Yep. Nitrate in soils, if you measure in a nitrogen-limited system, most of the nitrogen is going to get taken up or consumed or recycled as ammonium, and you'd expect very little nitrate, okay, or very little nitrification. Nitrate concentrations in soils tend to be much higher in soils that have lots of nitrogen. Okay, so there's an excess supply of ammonium, so nitrification rates can get quite high. And then once you have nitrate production in soils, a negatively charged anion, very little capacity to hold on to that, and that tends to get leached really quickly. You guys have probably seen in agricultural systems, they sometimes leave buffer zones between streams and fields. Part of that is about this. So if you do have lots of nitrogen applications on fields, and you're creating lots of nitrate, we've actually figured out, just in the same way that our wastewater treatment plants, we've figured out how to maximize the conditions under which these organisms take nitrate as waste and turn it back into an atmospheric gas. Those riparian areas are also very effective as places where denitrification takes place or is favored. So what's denitrification?
Reduction.
It's reducing nitrate back to atmospheric N2 or back to N2O. So it's closing the nitrogen cycle. So it's taking nitrate and either converting it sometimes to NO, but mostly to N2 or N2O. Okay, and right back up here. Right, so it's the returning reactive nitrogen back into an unreactive form. Okay, and these are all microbial processes. There's actually, and I would have said a month ago, that they're all bacterial processes. There's some evidence that there's actually fungi that can do denitrification. I just learned about this literally a month ago. But most of this is bacterially driven processes. And what's important in denitrification is what is the proportion of N2 versus N2O. Obviously, this is an inert atmospheric gas. This is a potent greenhouse gas, and so.
What determines
Great question.
that equation?
It's the same stuff we've been talking about. Let's make some guesses. So, what's the difference between N2O and N2 chemically?
They're in different reduction states. So it probably depends, at least to some degree, on what the redox state of the environment is.
Okay, so which one would you suspect is more favored under low redox or really, really low oxygen concentrations?
Dinitrogen,
Why?
because it's not there to be accepted.
Doesn't have O, right?
Mm.
If you're making N2O, you gotta have O to make an N2O, right? And so that's a little bit of an oversimplification because there's oxygen in nitrate. But ultimately, what seems to regulate this is whether it's an oxic condition or anoxic conditions. And what seems to be important there is how saturated the system is. So when you have saturated systems, you tend to get way more N2 produced. And when you have and this is more common from soils than it is from streams, for example, right? When you have, it's water-filled pore space, they call it. So what's the soil moisture of the system? And if you have lower soil moisture, you tend to see more N2O. When you have more saturated systems, you tend to see N2. Having said that, like if we went and measured, people have done this, like from wastewater treatment plants, there's huge amounts of N2O coming off of wastewater treatment plants, but nothing like you would predict. As a proportion of the total N being denitrified, it's actually quite small. Most of it's coming out as N2. But that's one. There's other things that, like this process is really sensitive to pH, and that's probably because the organisms, there's actually just two very small, relatively small genera of bacteria that are doing this, and they just seem to be really sensitive to pH. And they have an optimum. So when you get really low pH, you shut down rates of nitrification. or high pH for that matter. Like in feedlots, you tend to suppress nitrogen fixation because it's outside of their pH range, and that's why you can get what I didn't show on here. Oh, it's right here, right? So if you're getting buildup of ammonium that's not being nitrified, then you can get volatilization. So that's the feedlot manure thing, right? So high pH, it's outside the range of optimum nitrification, and so you get lots of ammonia volatilization.
What happens to N2O in the atmosphere? It's just float around for a long time?
Good question. Did you do your homework from last
I
time?
forgot.
What was it?
Leaf longevity.
Yeah.
Needle longevity. I just remember when you brought it up.
3 to 5. We settled on 3 to 5.
Okay.
I'm guessing 3 to 5, right? Did we say 3 to 5?
I think there's, I Googled it real quick, and I think there's longer lifespans.
Yeah.
I think that's like 20 years.
Okay.
Bristlecone was what came up.
Most of the nitrogen compounds in the atmosphere get degraded eventually chemically or like abiotically. So this one is really interesting. NOx. So NOx is common constituent of smog, and this in the presence of oxygen, which the atmosphere has lots of, and high radiation, so high ultraviolet light. This is actually a precursor to ozone. So NOx emissions are linked to tropospheric ozone, right? So it's that's just city smog. But in the process of that one gets broken down photochemically. I think N2O is the same. I think it takes a lot longer. I'm not sure what the atmospheric lifetime of N2O is, but I think it's something on the order of 20 years. Okay, so this is why this one, if you think about potent greenhouse gases, a lot of it just has to do with how long they stick around in the atmosphere. Methane, for example, gets degraded photochemically as well. So, gets scavenged by oxidants in the atmosphere in the same way that our body kind of has chemicals that scavenge that has, what is it, you take antioxidants or whatever. It's the same sort of thing. So there's chemicals in the atmosphere that scavenge these and break them down. But if there's much higher concentrations, like OH radicals in the atmosphere, they're called, break down a lot of the terpenes and isoprenes, and they just scavenge them and basically oxidize them in high light. And I would suspect, I don't know for sure, but I think N2O is the same.
So climate or increasing drought in some places would theoretically change the ratio of
N2 to
N2O
N2O?
versus N2.
Well, mostly you
Or is it
don't
not
see
that
much
sensitive?
drought. You don't see much denitrification in drier systems. The caveat of that is that most denitrification in upland soils is actually happening inside aggregates. And so you can actually create these biogeochemical gradients on really small scales where oxygen values decrease and where water changes. And so if you're measuring bulk N2O production from soils, for example, most of it's probably happening in really tiny aggregates inside of there. To say that drought is going to create more N2O than N2 seems a bit like I don't know that I would say that, but certainly if you let's say you have dry, and this has been, people have shown this in really cool ways in agroecosystems in Mexico, where you have long periods of drought and then you add a lot of water, like through a big episodic rainfall event, you get massive pulses of N2 out of those systems. But mostly N2O production, just background N2O production from soils is really low. Most of the big N2O production and increase in N2O that we're seeing in the atmosphere is in agricultural systems, from wastewater treatment plants. The biggest natural source of N2O production in just natural systems is the tropics, right? Because there's lots of nitrogen, it's wet, it's warm, right? And so lots of nitrogen cycling around in excess of what plant demand is, and so that's where you tend to see high rates of denitrification. The other part of that is I showed you kind of a budget of, maybe I didn't show you this. Yes, I did. I showed you a graph a couple of weeks ago that showed how much fertilizer we're adding in in the northeast watershed or across watersheds globally. So how much N we're adding versus how much is coming out in streams. And so the difference of that, a small proportion of that is actually being taken up, but it's thought that most of the nitrogen is actually being denitrified, and that's probably happening in riparian areas and in streams. Okay? So wherever you have a lot of water, and this, remember when we talked about, well, I remember no one took a stab at this question, right? But I showed you how the elements or the compounds and how they decrease over time after you flood a soil. N2O, denitrification, is just down from respiration, carbon or oxygenic respiration on the energy chain. And so this is a anaerobic process, right? And it's only really happening either in anoxic microsites or in environments that are anoxic. So wetlands, riparian areas that are saturated, those are where most of the denitrification is happening. Ben, did you have a question? Oh, okay. Okay, so just really briefly, nitrogen mineralization, we're taking organic N, we're turning it to ammonium. This is organically bound N, so things that are like, proteins. Okay, and we're decomposing that and turning it into a mineral form. So just recognize the difference between organic nitrogen and mineral nitrogen. This happens as a part of decomposition. A lot of decomposition is actually thought to be more about getting nitrogen than it is even about getting carbon, because there's lots of carbon, right? And most of that is just being respired. It's not being assimilated into biomass. Yeah?
Are they only reactive forms of nitrogen in mineral forms?
Yes, I'm thinking about that. So, organic nitrogen is reactive in the sense that it can be volatilized. No, that's not true, but it has to be mineralized into ammonium and then volatilized. Yeah, I think that's fair. So, yeah, the distinction being, usually when I'm talking about mineral N, I'm talking about one of these two, right? So they're just like a nutrient as opposed to something that's already stuck or bonded still to organic matter. Okay, but this is the first step of turning organic nitrogen into a form of nitrogen that most plants can take up. This is enzymes. Okay, more enzymes involved with this in nitrogen mineralization. Okay, just decomposition enzymes. Immobilization, we're taking ammonium or nitrate and it's coming back and getting turned back into organic nitrogen, so assimilated into organic matter. And I say here, immobilization is often dominant when C to N substrates is greater than 20 to 30. So if you have lots of carbon and very little nitrogen, then this is when you'd expect high rates of immobilization. Okay, because the supply of nitrogen is really low, and so when it does become available through mineralization, it immediately gets immobilized. And people have looked at this, another clever use of tracers, isotopic tracers. We followed this stuff around. I'll finish up and tell you more about that next time.